Imagine you and your friend are eating M&Ms and you have two large M&Ms stuck together within your packet, whereas your friend's M&Ms had a white colored one. Are these things regular for this type of candy? No, these are errors that have occurred during the manufacturing process. This is exactly how errors in DNA replication within living beings occur. These are called mutations. A mutation is defined as a permanent change in the genetic material of an organism. All mutations are heritable in that they will be copied during DNA replication, but not all mutations will be passed on.
Let's take a step back and look at how evolution contributed to the rise of mutations. The vast majority of mutations either have no effect or are harmful, but a very small percentage can be beneficial. The balance between complete fidelity and accuracy of DNA replication and low mutation rates has allowed the evolution of the rich diversity of species we see on Earth today. Early on, Mendel described genetic traits to come from specific genes. He thought that genes are unchanged and passed down generations. He realized that even if some genes do not appear in one generation, it can be found in the next. We know that this is the general rule to find patterns of inheritance. However, if this rule was always true, there would be no genetic diversity or change. And this is where mutations come in.
Mutations alone is a very broad concept. So let's focus on mutations that can alter single gene functions, which are small-scale mutations. A point mutation is a chemical change that affects just one or a few nucleotides. It can involve the substitution of one nucleotide for another, or even inserting or deleting one or more nucleotides. If a point mutation occurs in a gamete or in a cell that gives rise to gametes, it may be transmitted to offspring and to future generations. If the mutation has an adverse effect on the phenotype of a person, the mutant condition is referred to as a genetic disorder or a hereditary disease. For example, tracing the genetic basis of sickle cell disease to the mutation of a single nucleotide pair in the gene that encodes the beta-globin polypeptide of the hemoglobin. Now, let's dive into the nucleotide substitutions a bit more, which is the replacement of one nucleotide by another, and may have a relatively minor effect on the metabolism of the cell. They can be divided into three different types: silent, missense, and nonsense. A silent mutation is when a substitution has no effect on the encoded protein due to the redundancy of the genetic code. In other words, a change in a nucleotide pair may transform one codon into another that results in the same amino acid. Then there's missense mutations, which can be divided into two more specific categories: conservative and non-conservative missense mutations. A conservative missense mutation is where the resultant amino acid is the same or has similar properties to the substituted amino acid. For example, if a hydrophobic amino acid is replaced by another hydrophobic amino acid, it is highly unlikely that this substitution will have any effect on the protein structure and function. Therefore, since there will be little to no change as a result of the mutation, this would be classified as a conservative missense mutation. A non-conservative missense mutation is where the resultant amino acid has different properties from the original amino acid. For example, when a non-polar amino acid is substituted for a charged polar amino acid, this will most likely result in a change in the protein's shape and function. This is in fact the case in sickle cell anemia, where valine, a non-polar and hydrophobic amino acid, is replaced by glutamic acid, a charged and acidic amino acid. This substitution results in a fundamental change in the shape of hemoglobin and is the primary contributor to the pathology associated with sickle cell anemia. The final category is nonsense mutations, where a point mutation changes a codon from an amino acid into a stop codon. This causes translation to be terminated prematurely, and the resulting polypeptide will be shorter than the intended polypeptide encoded by the normal gene. Nearly all nonsense mutations lead to non-functional proteins. Then we have frame shift mutations, or when nucleotides are inserted or deleted, which can have a much more disastrous effect on the resulting protein compared to substitutions. They alter the reading frame of the genetic message, known as the triplet grouping of nucleotides on the mRNA that is read during translation. All nucleotides downstream of deletion or insertion will be improperly grouped into codons. The result will be extensive missense, usually ending sooner or later in nonsense and premature termination. And similar to nonsense mutations, unless the frame shift is very near the end of the gene, the protein is almost certain to be non-functional. Sometimes frameshift mutations can even cancel each other out with the addition of one nucleotide at one place on one gene, making up for the deletion of another nucleotide further along the coding sequence. Ultimately, however, this can still result in a missense mutation and affect the overall code and protein produced.
Now that we've covered small-scale mutations, let's move on to large-scale mutations that can have a severe impact on large chromosomal regions. Also known as chromosomal mutations, these can involve rearrangements of genetic material that affects multiple genes, including genes located on separate chromosomes. Errors in meiosis or damaging agents such as radiation can cause a breakage of a chromosome, which can lead to 4 types of changes in the entire chromosome structure. The first change is deletion, which occurs when a chromosomal fragment is lost. The affected chromosome is then missing certain genes. Within this scenario, duplication can also occur, where the deleted fragment may become attached as an extra segment to a sister chromatid. Alternatively, a detached fragment could attach to a non-sister chromatid of a homologous chromosome. In that case, though, the duplicated segments might not be identical, because the homologs could carry different alleles for certain genes. Deletions and duplications are especially likely to occur during meiosis. In crossing over, non-sister chromatids sometimes exchange unequal-sized segments of DNA, so that one partner gives up more genes than it receives. The products of such an unequal crossover turns into one chromosome with a deletion and one chromosome with a duplication. A chromosomal fragment may also reattach to the original chromosome, but in the reverse orientation, producing the third type of change, an inversion. Finally, a fourth possible result of chromosomal breakage is for the fragment to join a non-homologous chromosome, known as translocation. In reciprocal translocation, the most common type, non-homologous chromosomes exchange fragments. Here, the balance of genes is not abnormal. All genes are present in their normal doses. Nevertheless, translocations and inversions can alter the phenotype because a gene's expression can be influenced by its location among neighboring genes. Now let's talk about transposable elements, which are stretches in DNA in prokaryotes and eukaryotes that can move from one location to another within the genome. Even though they're also known as jumping genes, they never completely detach from the cell's DNA. Instead, the original and new DNA sites are brought very close together by enzymes and other proteins that bend the DNA. It often causes insertional mutagenesis, the creation of mutations in DNA by the addition of one or more base pairs, by disrupting gene functions, inducing chromosome rearrangements, or altering gene expression. These insertions can cause diseases or provide genetic variation for adaptation. A transposon itself is a eukaryotic transposable element which moves within a genome by means of a DNA intermediate. Transposons can move by a cut and paste mechanism, which removes the element from the original site, or by a copy and paste mechanism, which leaves a copy behind. Both of these mechanisms require an enzyme called transposase, which is generally encoded by the transposon. Removing or replacing a transposon from one place to another can interrupt the function of a gene or separate a promoter sequence from the gene it controls. Think of a promoter sequence as the on or off switch for a particular gene. We'll end off this section with retrotransposons, which are eukaryotic transposable elements that move by means of an RNA intermediate that is the transcript of the retrotransposon DNA. Retrotransposons always leave a copy at the original site during transposition. In order to be inserted at another site, the RNA intermediate is first converted back to DNA by reverse transcriptase, an enzyme encoded by the retrotransposon, but they can also be encoded by retroviruses. In fact, most transposable elements in eukaryotic genomes are of this type.
Now that we have covered the different types of mutations, we must understand how these mutations actually occur. When we're looking at spontaneous mutations, many occur due to the molecular interactions that occur naturally within the cell. If an incorrect nucleotide is added to a growing chain during replication, the base on that nucleotide will then be mismatched with the nucleotide base on the other strand. In many cases, the error will be corrected by DNA proofreading and repair systems. Otherwise, the incorrect base will be used as a template in the next round of replication, resulting in a mutation. While incorrect base pairing by DNA polymerase during DNA replication occurs, the rate of spontaneous mutations depends on the organisms and the different genes with a single cell. Although every cell undergoes spontaneous mutations, certain environmental factors can increase the rate of mutation. Mutations that are caused by agents outside the cell are called induced mutations. Now, a substance or event that increases the rate of mutation in an organism is known as a mutagen, which includes a number of physical and chemical agents that interact with DNA in ways that cause mutations. There are two types of mutagens, chemical and physical mutagens, both of which we will get into later during the presentation. Another cause comes from cumulative mutations. They are a series of spontaneous and induced mutations that can add up to more serious damage. Most cancers occur due to a combination of mutations. Some could be inherited, while others may arise from mutagens in the environment. Mutations accumulate within a cell, which is why exposure to chemical mutagens over time increases the risk of developing cancer.
Now that we have taken a closer look at the causes of mutations, we may wonder, if mutations all remained intact, what would happen to your body? Well, the cumulative effect on your body would be severely harmful. However, the cells produce many enzymes that constantly repair the damage to your DNA. There are many repair pathways that allow a cell to recognize and act on different types of mutations, but we will be looking at three specific repair mechanisms: direct repair, excision repair, and recombination repair. Firstly, direct repair is when a cell is able to reverse damage to its DNA. For example, the proofreading function of DNA polymers during DNA replication identifies and corrects mismatched nucleotides. Another example of direct repair is when bacteria, specifically E. coli, produces an enzyme that can break pyrimidine dimers rather quickly. As direct repair undoes the damage to the DNA molecule, it is different from a repair process that excises and replaces the damaged section. Most cellular systems for replacing incorrectly paired nucleotides, whether they are due to DNA damage or to replication errors, use a mechanism that takes advantage of the base paired structure of DNA. Yet, in many cases, an excision repair mechanism occurs when a damaged section of DNA is recognized and replaced by a newly synthesized correct copy. This type of repair involves three steps. First, a repair enzyme, often nuclease, recognizes an incorrectly paired base or distortion in the double helix and cuts out the damaged section. Then the DNA polymer synthesizes a new section by using the undamaged DNA strand as a template. Finally, the DNA ligase seals the new section in place along the molecule. Sometimes mismatched nucleotides evade proofreading by a DNA polymers. During this process, there are specific mismatch repair enzymes that prevent these replication errors from happening. Other enzymes tend to remove and replace incorrectly paired nucleotides that have resulted in replication errors. The difference between mismatch repair enzymes and DNA polymers is that while DNA polymers adds nucleotides and proofreads during replication, mismatched repair enzymes operate after replication to fix errors that have escaped proofreading. Now, what if a mutation causes damage to both strands? There is no template to build a replacement section of DNA in this case. When this occurs in a somatic cell, it undergoes a recombination repair mechanism. Recombination repair is when enzymes in the somatic cell may use a homologous portion of a sister chromatid as the template to construct a new DNA. However, this is most likely to contain a few errors, but it is much less damaging than if there were to be no repair that took place at all. If DNA damage is too severe to fix, the cell may perform triggered suicide rather than repairing itself. Once they die, it prevents the mutation from being passed on to daughter cells. So, now you've mastered the types, causes, and repair mechanisms behind mutations. That'll wrap it up for errors in DNA replication, and we'll pick it up from the presentation. Thanks for watching.